Hello again. This next lecture is going to be a bit of a mix of different topics. We're going to start off by looking at how you can use probability theory in order to predict the outcome in terms of the offspring that result from a cross between two different genotypes. We'll look at both predicting outcomes for a single gene situation and for two gene situations. We're also going to look at what a test cross is and how you use it. And then we're going to look at situations where you're doing crosses while looking at more than one gene, and we're going to focus on two genes. Finally, we'll touch on briefly a little bit of human genetics just for fun. Well, off we go. In the last lecture, we introduced the monohybrid cross, and we talked about how one could predict the outcome of crosses involving a single gene by using a Punnett square, as shown here. However, you can also predict the outcome of crosses by using probability theory. Applying probability theory for genetic crosses is pretty straightforward and involves only two rules. The product rule, which is applied when you're trying to work out the probability that two independent events will both occur, meaning the probability that both event A and event B will occur, typically simultaneously at the same time. When you're dealing with these types of probabilities, you multiply the probability that event A will occur by the probability that event B will occur. In the case of the sum rule, it is applied when you're dealing with the probability of two or more mutually exclusive events producing the same outcome. For example, you're trying to figure out the probability of either event A occurring or event B occurring. They can't both happen at the same time. Either one will happen or the other in order to give a particular outcome. When you're dealing with probabilities of A or B occurring, then you add the probabilities. Let's give a simple example. Let's begin by looking at rolling a single die. If you were to roll a single die, what would be the probability of rolling a 2? Well, this die has 6 sides, and each side has a different number associated with it. Given that you've got 6 different options, and the likelihood of any one of them showing up on top is a random event, there's a 1 out of 6 probability that 2 will show up on top. Let's now look at a situation where we're going to be rolling 2 dice. What would be the probability of rolling a 2 with the first die and rolling another 2 with the second die? So here we're looking at trying to work out the probability of 2 different events happening essentially at the same time. They're independent events. The first event is rolling a 2 with the first die, and the second event is rolling a 2 with the second die. We want to know the probability of both these things happening, so we're connecting them with an AND. Which rule are we going to use in this situation? I hope you can recognize the product rule here. The probability of rolling a 2 with the first die is 1 out of 6, and the probability of rolling a 2 with the second die is 1 out of 6. The probability of rolling these both at the same time together will be 1 out of 6 times 1 out of 6, which is equal to 1 out of 36. Let's look at a slightly more complicated situation now. So please pause the video and see if you can work out the probability for this particular situation. Now, how would you go about approaching this problem? Well, first of all, we want to look at the probability of rolling a 6. And if you think about 2 dice, there are lots of ways in which you could roll those 2 dice in order to come up with 6. So you have to start by thinking about how many different ways you can get a total of 6 on the dice. And again, you might want to pause this video at any moment to start working on this yourself. This is going to be useful to you in terms of actually understanding the principles. One way to get a total of 6 is to roll a 1 on the first die and a 5 on the second die. The probability of rolling these two is equal to 1 out of 36 because we're looking at using the product rule here. It's one event and the second event. We could also roll the 2 die and get 5 on the first die and 1 on the second die to get a total of 6. The probability is also 1 out of 36. Similarly, one could roll a 2 and a 4, or a 4 and a 2. One could also roll 2 3s, so both the first die and the second die would roll 3. That would give us a total of 6, and here's our probability. How do we figure out the total probability of any one of these happening? In each individual case, we've been using the product rule, but now we want to look at the possibility of rolling either 1 and 5, or 5 and 1, or 2 and 4, or 4 and 2, or 3 and 3. So what rule do we need to use now? You can look at these two possible rules and figure out which one you think should apply. As we're using OR, this is indicating that we're dealing with mutually exclusive events. These can't all happen at the same time if you're only rolling 2 dice. So this tells us that the sum rule is going to be applying, and in order to get the probability of rolling a 6 on the 2 dice, we have to add all of these probabilities. So the probability of rolling 6 on the dice when you roll 2 dice is actually 5 out of 36. You can apply these same general principles when you're dealing with genetic crosses. So if we come back to our simple single gene monohybrid cross, as shown here for the seed-shaped gene, and we can begin by calculating the probability that the zygote will receive the 2 dominant alleles, one from each parent. The probability of receiving the round allele from either of these parents is 1/2, because half of the gametes in each case will have a round allele and half of the gametes will have a wrinkled allele. Thus, we're looking at applying the product rule, 1/2 times 1/2, because we're looking at receiving the round allele from one parent and the round allele from the other parent, and so the probability is 1/4. Similarly, if you're looking for the probability of 2 wrinkled alleles in this cross, it's going to equal 1/4. But what about the probability of having an offspring that is a heterozygote? Well, in this case, you can get a heterozygote in 2 different ways. You might receive the round allele from the father and the wrinkled allele from the mother, or you might receive the wrinkled allele from the father and the round allele from the mother. In both cases, the probability is 1/4. So to find out the probability of forming a heterozygote, it can be formed either by receiving the round allele from the father or by receiving the wrinkled allele from the father. This is an OR event, so in order to get the overall probability, you're going to use the sum rule and add those 2 probabilities. So 1/4 plus 1/4 equals 1/2. Thus, there's a 50% chance that the offspring will be heterozygous. Here we've been generating the probabilities for the genotypes of the offsprings. We can equally generate probabilities for the phenotypes of the offsprings. When working out the probability of having round seeds in the offspring, you're looking at 2 different ways in which you can get round seeds. The first is having a genotype which is homozygous round, or having the heterozygous genotype. Both of these will yield the same phenotype. So we're really looking for the probability of event A, homozygous round, or event B, heterozygote, occurring. Since we're looking at either or, these are mutually exclusive outcomes, we're going to use the sum rule and add the different probabilities. So the probability of homozygous round is 1/4, and the probability of heterozygote is 1/2, and that yields an overall probability of 3 out of 4. Similarly, if you're trying to work out the probability of having wrinkled seeds in the offspring, it's going to equal the probability of only one possible outcome in the genotype, and that is homozygous recessive. That probability is 1/4. This yields you the 3-to-1 ratio in the phenotype, which is what Mendel saw when he did his monohybrid crosses. Now you might say, well, I can just use the Punnett square and get the same outcome just as fast and maybe faster, and that is true when we're dealing with a single gene. But when you start to work with probabilities of crosses that involve 2 or 3 or 4 genes, using the Punnett square becomes increasingly complicated and difficult and time-consuming. And we'll come back to this a little bit later in the lecture. Let's now turn to the situation where you have an organism that is showing the dominant phenotype, and you want to know whether or not that organism is actually a homozygous dominant or heterozygous, because both will yield the same dominant phenotype. In the absence of molecular genetics, so let's say you can't use PCR to identify specific alleles, how will you figure out the genotype of this organism? The simple way to do this is to use what's known as the test cross, and this is how the genotypes of such individuals were identified for a very long time until the development of molecular genetics. In order to determine if an individual with a dominant phenotype is a heterozygote or a homozygote, you simply cross that individual with the homozygous recessive. The outcome in terms of what the offspring will look like will depend on whether or not the unknown individual, which is the individual with the dominant phenotype, is homozygous dominant or heterozygous. If it's homozygous dominant, you'll see the outcome to the left of the slide. In this case, all of the offspring will have the dominant phenotype. If the unknown is a heterozygote, then half of the offspring will have the dominant phenotype and half of the offspring will have the recessive phenotype. And if this doesn't make sense to you, just work out the cross on your own. What happens when you're dealing with more than one gene? For example, 2 genes, and you want to predict the possible outcomes in the offspring. Let's assume that the 2 genes are on different chromosomes and that those 2 chromosomes are assorting independently. And we'll work with different characteristics of the P again. In this case, we're looking at P shape and P color. P shape, as you know, has 2 alleles, one that codes for a round shape and one that codes for a wrinkled shape. P color can also vary. It's controlled by a different gene, and it has 2 alleles, one that codes for yellow and one that codes for green peas. And the yellow allele is dominant over the green allele. Mendel studied this same type of a cross, beginning first with 2 pure breeding parent varieties, one that showed dominant phenotype for both of these genes, and crossed it with a pure breeding variety that was homozygous recessive for both genes. The offspring would all be heterozygous, and I hope you can see why. Keep in mind that each gamete has to receive 1 allele for each gene. The next big question for Mendel was, what happens if I allow these heterozygotes to cross-fertilize each other? Well, to picture what's happening inside the germline cells of these individuals, we have to go back and think about chromosomes. And we're now talking about chromosomes in cells that are about to undergo meiosis. So here we're just going to consider the 2 chromosomes that are involved in this particular cross. This is the cell from the heterozygote F1. So one of these chromosomes has the dominant allele and one of these chromosomes has the recessive allele. The recessive allele came from the mother over here, and the dominant allele came from the father over here. Similarly, there's the second chromosome which carries the gene for P color. One of those chromosomes has the dominant allele, it came from the father, and one of these chromosomes carries the recessive allele, it came from the mother. These chromosomes now have to be assorted in meiosis. During meiosis, the 2 homologous chromosomes will be pairing up in prophase 1, and then they'll be separated during anaphase 1. How will these different chromosomes be separated from each other? Well, it depends on how they all land at the metaphase plate. One option shown to the left-hand side is that the chromosomes will align at the metaphase plate such that the chromosomes with the 2 dominant alleles, the dominant round allele and the dominant yellow allele, will land on the same side of the metaphase plate. And the chromosomes that are carrying the recessive alleles for both of these genes will land on the other side of the metaphase plate. The result are 2 daughter cells, one that has both recessive alleles and one that has both dominant alleles. In the second meiotic division, the sister chromatids will be separated, so the outcome will be half of the gametes from this type of a division will have the 2 dominant alleles, and half of the gametes of this division will have the 2 recessive alleles. However, these chromosomes could have landed on the metaphase plate in a different orientation, and this is diagrammed on the right-hand side of the slide. Here, the chromosomes have landed at the metaphase plate such that the dominant allele for the seed shape gene is on the same side as the chromosome with the recessive allele for the seed color gene, shown here. Similarly, the chromosome with the recessive alleles for seed shape landed on the same side as the chromosome with the dominant alleles for seed color. When these are separated in anaphase 1 of meiosis, it generates two daughter cells, one that has the dominant allele for seed shape with the recessive allele for seed color, and a daughter cell that has the recessive allele for seed shape and the dominant allele for seed color. These sister chromatids are separated in meiosis 2, and they yield daughter cells, half of which have the dominant allele for seed shape and the recessive allele for seed color, and half which have the recessive allele for seed shape and the dominant allele for seed color. These are the four possible outcomes that you can get in the gametes, and each outcome is equally likely to happen, so we end up with four different kinds of gametes. One type which has both dominant alleles, one that have both recessive alleles, one that has dominant seed shape and recessive seed color, and one that has recessive seed shape and dominant seed color. Each one of these possible gamete outcomes is equally likely to happen, so it's a quarter probability for each possible gamete type. Now we know what the possible gametes are, let's put them together in a Punnett square and figure out what the offspring might look like and the ratios of the offspring genotypes. So along one side of the Punnett square, you're going to put the possible gametes from one parent, in this case the male is on the side, and along the top you put all of the possible gametes for the female, the other parent. In each one of these grids, you're going to place both the alleles from the mother and the alleles from the father in order to generate the alleles or possible genotypes of the offspring. And all of this is depicted here. Each one of these grids now shows a possible genotype in the offspring. And because there are 16 grids, that means the probability of any one of these outcomes is 1 out of 16. If you look carefully, some of these outcomes are actually the same in terms of their genetic makeup, or the genotypes. By adding up the number of boxes that contain the same genotype, you can generate the overall probability for each different genotype in the F2 generation, and that's shown here. Once you have the genotypes, you can also use this grid to generate the probability of different phenotype options. So here we're looking at that same grid with all of the possible genotypes, each one of these representing 1 out of 16 as a probability, and then you simply identify which genotypes would yield the same phenotypes. So in the case of the two dominant phenotypes, meaning a round seed shape and a yellow seed color, we've got 1, 2, 3, 4, 5, 6, 7, 8, 9 of these grids or squares that will yield the same dominant phenotype. Thus, the probability of having F2 offspring that are round and yellow is equal to 9 out of 16. If we now look at an outcome where we have green seeds, the recessive phenotype for the seed color, but round shaped, the dominant phenotype for the seed shape, we find that there are 3 squares or grids that carry that phenotype. And thus the probability of round green seeds in the F2 generation is 3 out of 16. If we now look at the recessive seed shape, which is wrinkled, and the dominant seed color, which is yellow, we have 3 different squares that yields this phenotype, and thus the probability of a wrinkled yellow seed is 3 out of 16. And finally, looking at the double recessive phenotypes, we find 1. So the probability of wrinkled green seeds is 1 out of 16. If you now add up the different ratios for each one of these, it yields the 9 to 3 to 3 to 1 ratio, which is the classic dihybrid cross ratios in the F2 generation. Now, if you were to do this by yourself, and if you took the time, this could take you I don't know, maybe 20 minutes, maybe 30 minutes to actually probably 30 minutes to generate this big Punnett square and then figure out all the genotypes and the phenotypes. And you can see how it's kind of prone to making errors because if you miscount or don't see something, you can make a mistake. You can use probability to calculate these genotype and phenotype ratios much more quickly and with a lower likelihood of making a mistake. And you do this by considering each gene separately. If you want to get some practice at doing this, I've got a video that's already posted underneath this lecture which walks you through this process using something called branched diagrams. This is something that you'll be doing in second year genetics, so it's worth your while figuring it out. If you really hate probabilities and don't want to deal with this, you can always work out your probabilities for crosses involving two genes by generating Punnett squares, and that's fine. So for your homework this week, I'd like you to work out this problem, and if you're feeling very clever, you should first work out the problem using a Punnett square, and then try and generate the results using probability. We can take this up on Friday or on Monday. Now, Mendel was working with pea plants. Does any of this relate to human genetics, where they have a single gene with two alleles and a simple dominant recessive relationship? And the answer is yes, but phenotypes are not always the result of a single gene. Often you've got more than one gene which is contributing to a particular phenotype. But there are some exceptions. Often those exceptions that we know about in human genetics are associated with disease. For example, we've already talked about cystic fibrosis in this course, and you'll remember that the cystic fibrosis gene codes for a channel protein, shown here. And this channel protein allows chloride to move through it, so it's a chloride channel. The normal allele for this gene is a dominant allele, and it is dominant over alleles that cause cystic fibrosis. In other words, cystic fibrosis is a recessive disease, so individuals that have at least one copy of the normal allele, in other words, they're heterozygotes, will have a normal phenotype. Individuals must have two copies of the cystic fibrosis allele in order to have cystic fibrosis. What is the cystic fibrosis allele? It is an allele which codes for an abnormal chloride channel, which is non-functional. Now, the cystic fibrosis gene codes for a protein, this is the channel here, which is about 1,480 amino acids long. So not surprisingly, it's not just a single mutation that gives rise to the cystic fibrosis allele. There have been a number of different mutations identified, all of which yield abnormal protein or abnormal chloride channels. And that means that there's more than one allele that's responsible for cystic fibrosis. There is a specific allele which contains a mutation that results in the loss of a single amino acid, which is phenylalanine, in the protein. And this is the most common mutation one sees in cases of cystic fibrosis. But there are other mutations as well, meaning that there are other alleles or versions of this gene which also yield abnormal protein. One, for example, codes for a stop codon somewhere in the middle of the gene, which leads to a totally non-functional, small, truncated protein. Now, when we're working with human diseases and we talk about a disease which is due to a recessive allele, individuals who are heterozygotes and therefore have a normal phenotype are referred to as carriers because although the phenotype is normal, they're actually carrying the disease allele. So with that in mind, you should take a minute, pause the video, and work out this problem. We can take this up this Friday or on Monday. Some disease alleles can actually be dominant, and a nice example of this is the allele that's responsible for Huntington's disease. Huntington's disease is a progressive neurological disorder which is caused by a mutation in one gene which is on chromosome 4 of the human genome. Individuals with Huntington's disease usually start to develop symptoms as adults, and often a little bit later in life, so after 40, although it can come on at any time. And these symptoms will include uncontrolled movements, psychiatric problems, and a loss of cognitive function. It's a progressive disease, meaning that things get worse as the person gets older. Eventually, the disease result in degrees of disorder which cause death. The gene responsible for Huntington's codes for a protein. The protein is expressed, of course, in cells in the brain. The gene responsible for Huntington's codes for a protein that contains a series of glutamines, the amino acid glutamine, within it. And that's shown here. We've got this protein. These are the amino acids. Each circle is an amino acid. And somewhere in the middle, you'll have a series of glutamines all in a row. These glutamines are coded for in the messenger RNA and in the non-template section of the DNA as CAG repeats. So CAG, 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 CAG. And normal versions of the gene have 10 to 26 of these repeats, meaning there's going to be a little strip of 10 to 26 glutamines. Alleles that give rise to Huntington's disease have more of these repeats. They'll have on the order of 36 to 121 of these CAG repeats in the non-template strand of the DNA and the messenger RNA, coding for 36 to 121 glutamine amino acids in this protein. The presence of these extra glutamines seems to affect the ability of the protein to fold properly, and as a result, they have a shape which causes them to aggregate together. So the proteins all stick together. This disrupts the functioning of the cells, and this leads to the misfunctioning of the cells and the neurological disorder. The disease alleles are dominant alleles. So that means that even in heterozygotes, in the presence of a normal version of the gene or a normal allele, the presence of the abnormal allele is sufficient to generate the neurological disorder. Take a minute, pause this video, and try and think about what this means at the cell level. Both of these alleles should be expressing. That means you have about 50% of the protein being generated, which is normal in a heterozygote, and 50% which is abnormal in the heterozygote. And the presence of this abnormal protein is sufficient to cause the illness that we're seeing. This is because the abnormal proteins are aggregating together. It doesn't matter if there's some normal protein, the abnormal protein aggregating together is going to cause a disruption to the cell function. There are two different take-homes I think you can get from this. The first is that when we talk about normal allele versus disease allele, it doesn't mean there's necessarily a single allele which is normal and a single allele which is abnormal. In this case here, you can get any number of alleles which are normal as long as the number of glutamines being coded for is in the order of 10 to 26. You can also have a whole series of alleles which give rise to abnormal gene products or abnormal proteins, and in this case, any allele that has 36 to about 121 repeats is going to yield an abnormal protein. It's going to give rise to the disease. The second point that's being made here is that whether or not an allele is going to act as a dominant or a recessive allele has to do with the actual product it's coding for and how the presence of the abnormal product impacts the functioning of the cell. In the case of Huntington's disease, half of the protein being abnormal gives rise to the disease. In the case of cystic fibrosis, having half of the chloride channels functional in the cell seems to be sufficient to keep and maintain a normal phenotype. There's just so much more we could say about human genetics. I'm sure many of you are really interested in it, but we'll have to leave that for another course.